The amount of information from only one experiment is enormous. We can detect the presence of these cell wall polymers and know exactly how they are distributed in cells and tissues, and even determine their abundance.
This method is designed in such a way that it can be applied to almost any kind of plant tissue from any species one may want to analyze.
Immunolocalization on plant tissues involves a lot of meticulous work, difficult to explain without actually seeing what happens.
Before collecting the plant tissue sample, fill a glass vial with enough cold fixative solution to completely submerge all the samples. Then select the plant tissues to be analyzed, and trim the samples to a size of no more than 16 square millimeters. Immediately immerse the samples in the fixative solution.
Glutaraldehyde and formaldehyde are both excellent fixatives, but both are hazardous and must be handled in the flow hood.
Transfer the vial to a vacuum chamber and slowly apply vacuum. When the pressure reaches negative 60 kilopascals, the floating material in the vial will start to sink to the bottom. Maintain the pressure in the chamber at no more than negative 80 kilopascals. The sinking process may take up to 2 hours. After the samples have been in the vacuum chamber for 2 hours, slowly release the vacuum. Seal the glass vial and refrigerate it overnight at 4 degrees Celsius. After the overnight fixation, discard any samples that didn't sink to the bottom of the vial. Wash the remaining samples with 25 millimolar PBS for 10 minutes, and then wash them in 25 millimolar pipes buffer for 20 minutes.
It is extremely important to remove